So today's topic is Euchromatin and Heterochromatin. So first of all, let us know what does the word or term chromatin mean. Well, we know that in case of inside the nucleus, well, this is a cell. So inside the nucleus, there is chromatin, the less condensed form of the chromosome um, during the interphase. So and these and let's assume that this is the nucleus, and inside this there is certain dense regions where the density of these chromatin fibers is more. These regions are known as heterochromatin, and there are other regions which are less dense. So when stained, this heterochromatin part actually, well, let me write out here. So heterochromatin and euchromatin. So in case of heterochromatin, this is thickly stained. So since it is dense in this region, so it is thickly stained, and since it is its concentration or the amount of chromatin is spaced out, so it is lightly stained when one being stained. And how to remember this? Well, what does what the term U means? It means good. And when something is light or less dense, not that dense, we can see it quite properly, not appearing very blackish, so we can call it U or good chromatin, lightly stained. Or when, and hetero means different from the rest of the regions, so it is known as heterochromatin, which is darkly stained. And these lightly stained regions are, as I already said, that these are spaced out regions or quite open, and they are actually less condensed, and these heterochromatins are more condensed. That is the reason that they are thickly stained or highly stained. And well, let me fix the camera. And so the thing is that during transcription, if the chromatins are spaced out, only then the enzymes that are required for transcription, only then they can enter, right? So that is why euchromatins are transcriptionally active. These spaced out thing happens, these less condensed or spaced out euchromatins are out there, so that transcription can happen, or in these regions transcriptions do happen. And in case of euchromatins, transcription is suspended or do not happen, because they are not spaced out and they are condensed, so the transcription factors cannot easily enter them. Okay, and more points to be noted are these, that heterochromatins, they are actually replicated late in the S phase, late replication. Now, what can be the reason of that? A probable reason of that might be this, that since heterochromatins are not transcribed, so they are not that important to the cell, right? So it is replicated late, and then the thing is, they are actually the genetic recombination rate is actually, or frequency is actually low. Genetic recombination frequency is low. Now that can be explained with this, that since they do not yield any product, so there is not much requirement of genetic recombination to give out variations in the offsprings, because both would result in the same thing, right? And so that was it. And the thing is that heterochromatins can actually be divided into two types. Heterochromatin, one is the constitutive and the other one is facultative. Well, before this, I think we should do a recap thing. out here. We should recap it out. So euchromatin, right? So these are good, and they are transcriptionally active. And for being transcriptionally active, they require the chromatin to be less condensed. And since they are less condensed, so they are lightly stained. And in case of heterochromatin, they are not transcribed, so they can remain as condensed or more condensed forms. And since this is the thing, so they are thickly stained. And since they are not that important, I would not say that actually they are not important. For the case of remembering, you can remember in this way that since they are not that important, so they are replicated late. And genetic recombination rate is also low, since since their product since they do not give any product, so there is no variation variation in their genetic their primary structure or their genetic sequence is not that important for giving out variations. And now the thing is that heterochromatin can be divided into two types, that is constitutive heterochromatin and facultative heterochromatin. Well, constitutive heterochromatin, as the name suggests, this is actually those type of heterochromatins which are present in all cell types of our body. And in case of facultative, well, let us think one thing, that we are all made up, all of our cells are made up of the same genome, or the same genome. But we know that the cells of our eye, for example, the lens cells or the retinal cells, are vastly different from the from muscle cells, or from the neuronal cells, or from the heart cells or the cardiac cells. That is the thing. So what brings about these variations? Well, these variations are actually brought about by epigenetic changes. And so these epigenetic changes actually prevents the transcription of certain regions of the cells, and allows the certain regions of the genome and prevents the transcription of those regions in a different cell, so depending on the cell type. So in case of facultative, these type of chromatin materials are heterochromatin in certain cells, but they are actually euchromatin in a different cell. But in case of constitutive, they are actually heterochromatin in all the cells. Now the question is that if they are not expressed, this type of constitutive heterochromatin, then what is the need? Well, there is their need. Well, these chromatins, these constitutive heterochromatins, they are actually needed for the structure, for maintaining the structure or forming the chromosome. And these are actually repetitive. These sequences are actually repetitive. Well, that was it. And well, what I meant by structural, they structurally help. They actually provide the structure or provide structural stability to the chromosomes, these constitutive heterochromatins. And facultative, as I already said, an example of this facultative is bar body. Well, now if someone does not know what bar body is, well, we know that in case of human females, there are X and X chromosomes. There are 2 X chromosomes, and in case of male, there is 1 X chromosome. Now the thing is, here if 1 X chromosome is being expressed, and out here if 2 X chromosomes are being expressed, so this same thing is being expressed doubly, but here same thing is only 1, right? So since this is being expressed once out here, so either out here it has to be more expressed more. If that is the case, then excessive excessive of the product will be formed out here, since here 2 are there. Or here the expression of X would be less, and here the expression of X would be more, but that is not actually possible, because out in both cases the chromosomes are the same chromosomes, because this is also obtained from mother, and so this is from the mother, right? So the thing is that here, in case of a female, or where there are more than one X chromosomes, it sometimes there are 3 X chromosomes in certain disorders. So in those cases, only one chromosome remains transcriptionally active, and the rest of the chromosomes get transcriptionally inactivated, and this is called, these inactivated chromosomes are called bar bodies, and they are actually heterochromatins. They actually look highly stained or densely stained or thickly stained under the microscope, and these bodies, bar bodies are actually bar bodies. I have written it wrong. Bar, BARR, body. These bar bodies are actually facultative heterochromatins, because no one knows. I mean, for example, if from the zygote 2 cells are formed, and from that 4 cells are formed, ultimately in a mass of cells, certain cells might have these chromosome from the, certain cells, in certain cells the paternal X chromosome might be activated, and it's or inactivated, might form the bar body, and in certain the maternal ones might form the bar body. So in different X chromosomes are being inactivated or forming bar bodies. So this is also an example of facultative heterochromatin. So that was it. Thank you for watching.